Hello friends, welcome to Pariksha Mantra channel and subscribe my channel to get new videos. Today we see sterilization. This is important in microbiology, immunology and in any process. Sterilization word comes from Latin word sterilize, means unable to produce offspring or barren. Sterilization is a process by which all living cells, viable spores, virus and viroids are either destroyed or removed from an object or habitat. Sterilization is an essential stage in the processing of any product destined for parenteral administration or for contact with broken skin, mucosal surfaces, or internal organs where the threat of infection exists. Sterilization of microbiological materials, soil dressings, surgical dressings, ligatures, and other items is necessary to minimize health hazards associated with use of these. If they are contaminated with microbes, the products or material labeled sterile should be completely free from microbes or any kind of living cells. There are four main methods of sterilization, that is physical method, chemical method, radiation method and mechanical method of sterilization. We see first physical method of sterilization, in which two types of physical methods are there: dry heat sterilization and moist heat sterilization. There are some methods of dry heat sterilization, which are sunlight, red heat, direct flame, hot air oven and infrared radiations. Then we see dry heat sterilization. Heat is most reliable and rapid method of sterilization. The killing effect of dry heat is elevated due to proton denature, oxidative damage, and the toxic effects are elevated. Then we see first method of dry heat sterilization, that is sunlight and drying. This is natural method for sterilization of water in tanks, river and lakes. Sunlight possesses ultraviolet rays which along with heat rays responsible for appreciable germicidal activities. Then we see red heat. It is used to sterilize metallic objects by holding them on flame till they are red hot. Examples are needles, tips of forceps, inoculating wires. Then we see flaming. The flaming substance is passed over flame without allowing it to become red hot. Examples are cover slip, glass slide and vegetative microorganisms. Then we see hot air oven. This is most widely used method of sterilization by using dry heat. The principle of hot air oven is, in hot air oven, heat is transferred from the source to the load by the way of convection, conduction and radiation. The killing of microorganisms by heat is function of the time temperature combination used. If the temperature is increased, then the time required for killing all bacteria will be decreased. Then we see construction of hot air oven. In hot air oven, maintain the hot air high temperature. It consists of the double wall chamber of aluminum or stainless steel. Heat is provided by electric heating elements. Then we see diagram of hot air sterilization. Here we see the diagram. Then working of hot air oven. Non-heatable substance put inside the hot air oven. Then normally the space of bacteria as well as microorganisms are killed. The oven must be allowed to cool slowly for about 2 minutes before the open. The relation between temperature and time of hot air oven. The temperature, if 170 degrees Celsius, then time in minutes 60 minutes. If temperature is 160 degrees Celsius, then time required is 720 minutes. If temperature is 150 degrees Celsius, 150 degrees Celsius, then time required for it is 150 minutes. If temperature is 140 degrees Celsius, then time required for it is 180 minutes. Then we see advantages. It is simple and easy to use. It do not release any harmful flames. And applications of hot air oven is, it is used to sterilize glassware, forceps, etc. Also for some pharmaceutical substances. Then we see moist heat sterilization. In the process of microorganisms and killed by hot water steam, it kill the microorganisms by denaturation and coagulation of microorganisms. Moist sterilization divided into the three parts, that is temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, and temperature above 100 degrees Celsius. First we see temperature below 100 degrees Celsius. In this, mostly heat liable fluids may be sterilized by heating at temperature below 100 degrees Celsius. Example of it is pasteurization of milk. Then second, we see temperature at 100 degrees Celsius. Boiling at 100 degrees Celsius for 10 to 30 minutes kill all vegetative bacteria and some bacterial species. Atmosphere of free steam is used to sterilize the culture media, which may be decomposed at 100 degrees Celsius. Then we see temperature above 100 degrees Celsius. Example of it is autoclave, in which saturated steam is produced under pressure. Here we see the diagram of autoclave. The procedure of autoclave is, the water is filled in a autoclave and material is to be sterilized is allowed inside the autoclave. Now maintain the temperature and pressure for sterilization according to then. The temperature is 115 to 118 degrees Celsius, then steam pressure is 10 and holding time is 30. We see it in table. Then advantages of autoclave is, it is very effective, it is quicker in heating up than exposed, it provides greater lethal action of moist heat. The disadvantages of autoclave is, it is not suitable for anhydrous material such as oil, fats, etc. Not suitable for heat sensitive substances. Here are some references. Thank you.